Hello, physiology people. Today we're going to start chapter 6, muscle physiology. So I'm going to share my screen and we're going to get started. Okay, so this chapter is going to be broken up into about 4 lectures, and this is lecture 1. So what we're going to get through today is a quick review of muscle anatomy and different muscle types of the body, a little mention of the different types of muscle fibers that we have, and then we'll get into the sliding filament theory of how muscles contract. So let's begin. So this is review of chapter 4, part 1, and chapter 4, part 3, which is how skeletal muscles are stimulated to contract versus cardiac muscle versus smooth muscle. So let's start over here with skeletal muscle, which is voluntary muscle. We can control that consciously. So skeletal muscle involves the neurotransmitter acetylcholine, abbreviated ACH. Now, the receptor for that acetylcholine is called a nicotinic cholinergic receptor. And when I taught this to you in class, I said, think about trying to smoke a cigarette, which has nicotine in it. To pick up that cigarette and bring it to your mouth, it's going to take voluntary muscle action. So when you think nicotinic cholinergic, think as if you're picking up a cigarette, which is voluntary muscle movement. Okay, so that is always going to stimulate skeletal muscles to contract. Now, glycine and GABA are inhibitory neurotransmitters, and that causes muscles to relax. So this is also review. Okay, now we're going to compare the speed of contraction and whether or not they're prone to fatigue or fatigue resistant. So let's move on to cardiac muscle. So we don't have control over that. That is controlled autonomically under the parasympathetic system, which is rest and digest, and the sympathetic system, which is your fight flight. Okay, so parasympathetic neurotransmitter. During rest and digest, your heart rate should slow down. And the neurotransmitter that does that, again, is acetylcholine, because that's the neurotransmitter for that system. Now, the receptor for that acetylcholine is muscarinic cholinergic. So when you think muscarinic, think you must do this action because you have no control over it. So you must is involuntary, and that's a muscarinic cholinergic receptor. And when that neurotransmitter binds to that receptor, it's going to slow your heart rate down. Now, if it's fight-flight, sympathetic regulation, that neurotransmitter is epinephrine, also known as adrenaline. Its receptor is going to be an adrenergic receptor, but its specific one is going to be beta-1. So remember, how many hearts do you have? You got 1. So beta-1 adrenergic receptor. And if it's fight-flight, your heart rate's going to skyrocket. Okay, compared to skeletal muscle, which has the fastest contractile speed, the heart is only intermediate in its contractile speed. Your skeletal muscles are prone to fatigue and can give out, but your cardiac muscle is fatigue resistant because it never stops beating 24/7, 365 until it stops one day and then, well, you stop. Okay, so let's move over to smooth muscle. You don't have control over that as well, and it is regulated under the rest and digest parasympathetic system and the fight-flight sympathetic system. So for rest and digest, that's going to be acetylcholine with, again, muscarinic cholinergic receptors. Now, its fight-flight neurotransmitter is epinephrine again, but there are multiple adrenergic receptors, and I'm only showing a few of them right here. The beta-2 receptor on smooth muscle of your bronchioles and beta-2 receptor that's found on the smooth muscle of arteries going to skeletal muscle, and alpha adrenergic receptors that are found on all things GI. So your GI contractile speed is the slowest among the 3 categories here, but it's also fatigue resistant. So my question, which is review for my students, epinephrine binding to beta-2 adrenergic receptors causes what to happen? And the answer is your airways, your bronchioles open up wide so you get maximal airflow. And your skeletal muscles, the arteries supplying them with blood, those arteries vasodilate with that receptor as well. So remember, you have 2 lungs, the beta-2 receptor, And the beta 2 receptor is also best to get away. When you're running away from danger, your skeletal muscles need maximal blood flow. Okay, epinephrine binding to alpha adrenergic receptors causes all things GI shut down. That means peristalsis, smooth muscle, shuts down. GI glandular secretions, shuts down. Artery blood flow to the gut reduces, vasoconstricts, because GI activity isn't going to save you during fight flight. Okay, good review. So let's move on to muscle review. Okay, so everyone in anatomy has studied skeletal muscle organs, such as the biceps brachii or the triceps brachii or the hamstrings of the back of the leg or the quadricep muscle group on the front of the leg. But muscles are arranged like Russian nesting dolls. And if you know what they are, you open up a doll and inside that doll is a smaller doll. And you open up that doll and inside it is smaller doll. So skeletal muscle is arranged this way. You open up and look inside the muscle organ, and inside it's made up of muscle fascicles. All right, these little compartments are muscle fascicles. If you pull out a single muscle fascicle and you look deep inside, it's made up of multiple muscle fibers. And guess what? If you open up a muscle fiber, okay, that's a muscle cell. You're going to see that inside a fiber are basically many repeating units of muscle myofibrils. And each muscle myofibril has repeating units of sarcomeres. And the sarcomere is the functional unit of muscle contraction, meaning when muscles contract, they're contracting at the sarcomere level. Okay, so all the vocab for all of these is found on the next slide. Okay. So muscle organ is the largest structure of a muscle that you can see with your eyes. And then inside the muscle organ are a bundle of muscle fascicles. Inside a fascicle are a bundle of muscle fibers. That's the muscle cell. And by the way, the muscle cell is what the motor neuron is stimulating by releasing acetylcholine. And it's the muscle fibers that have acetylcholine receptors on them. Okay, so a single muscle cell or fiber is made up of many myofibrils, and each myofibril contains thousands upon thousands of sarcomeres. Okay, so that's just vocab. Muscle fiber types. Our muscles are made up of a mixture of these muscle fiber types. Some muscle groups contain more red or slow twitch fibers, and some might contain more white or fast twitch fibers. But the slow twitch fibers, the red fibers, are called that because, well, they have a pigment to them because there's more myoglobin and more mitochondria. So the dark fibers have muscle's own unique respiratory pigment called myoglobin, and it has a lot of blood supply, and it has many mitochondria for making a bunch of ATP so that skeletal muscles can function. They produce steady but little power. So these are your endurance muscles, like if you run a marathon or you play basketball or you run up and down a soccer field for an hour or two. These are the muscle fiber types that are going to be predominant in your core and in your legs. Okay? They fatigue slowly, but they can be pushed to their limits. But again, you'll find them in your leg and your core and posture muscles. Now, the opposite is white fibers or fast twitch fibers, which appear lighter in color because they have less myoglobin, slightly less blood supply, fewer mitochondria. And so these are the muscle fiber types that when you push them really hard, they're going to resort to anaerobic metabolism, produce lactic acid, and that precedes they will give out. So they produce short bursts of great power, but they can't keep that up for long. They're going to give out on you. So they will fatigue quickly. So your arm muscles and shoulder muscles would have more of these fiber types. Okay, so let's look at how a motor neuron stimulates a muscle cell. So we're looking at this thing right here, down here. It says a muscle fiber. That's a muscle cell. It has receptors on its cell membrane for acetylcholine. And this right here is a motor neuron that's going to stimulate it by releasing acetylcholine. That acetylcholine, if you look at the top image of here, crosses the synaptic cleft and then will bind to the receptor to stimulate that skeletal muscle. So if we take it step by step, the neuromuscular junction, where a motor neuron synapses onto a muscle cell. The somatic motor neuron is going to release the neurotransmitter, we just covered it on slide 1, acetylcholine. Now the receptor for that on skeletal muscles, remember, are voluntary. Think of the cigarette analogy. You have to voluntarily pick up a cigarette that contains nicotine to smoke it. So that receptor is going to be a nicotinic cholinergic receptor. When that receptor is open, sodium comes flooding in like a tsunami inside the muscle cell. And then the sodium, once it's in, stimulates the muscle cell, causes an action potential or an EPSB from chapter 1, so chapter 4, part 1. But the big thing, this action potential causes calcium to be released from a calcium warehouse in muscle cells called the sarcoplasmic reticulum. Okay. So let's look at the sarcomere's basic structure. A sarcomere contains these myofilaments called actin and myosin. Actin is the thin filament, and they show it as this purple string right here. Myosin, shown as the green structure here with little fingers on it, that's myosin. So I remember actin being the thin filament because if you stay active, you stay thin. Okay? Okay. Well, actin has active sites that myosin with those little fingers wants to grab and pull on actin. But there's going to be a problem. The problem is the protein called tropomyosin, the long T word, normally blocks these active sites so myosin can't grab it. So tropomyosin is attached to troponin, the short T word. Now the calcium that got released from the calcium warehouse with that action potential releases all this calcium. It comes flooding down at the sarcomere level. It binds to troponin. Then troponin moves, lifting tropomyosin off the active site on actin. Now with those sites exposed, myosin can grab actin and pull on it. And the pulling of actin against myosin is the sliding of the two filaments in the sliding filament theory. Okay, so let's go to the next slide and look at this. So here again is the motor neuron. It releases acetylcholine, shown here in the peach-colored synapse. Acetylcholine binds to its nicotinic cholinergic receptor, shown by this green thing right here. When that happens, sodium is released into the muscle cell. That causes an action potential. That action potential over here on the right travels deep down this T tubule, and it causes calcium right here, the little red balls, to be released from the shown in green sarcoplasmic reticulum. What calcium will do is bind to troponin on the sarcomere, on actin. It will cause tropomyosin to lift off the active sites. And when that happens, myosin can grab actin. So let's see what that looks like. Okay, step-by-step approach. The motor neuron releases acetylcholine. It binds to nicotinic cholinergic receptors on your skeletal muscles. That opens up sodium channels, and sodium floods into the cell, causing an action potential. The action potential enters the T tubule. That causes calcium to be released from the sarcoplasmic reticulum. Calcium then binds to troponin, and then tropomyosin lifts off the active sites, exposing them to the myosin fingers, and myosin readily grabs a hold of actin. And every place those little fingers make contact is called a cross bridge. And so it's just like tug of war. Myosin grabs actin and pulls on it. Okay? And it will continue doing that until the sarcomere itself shortens, and then the muscle cell will shorten, making the fascicles and muscle organ shorten. So let's look at this. Calcium is released, binds to troponin, the blue thing. Tropomyosin is the purple stringy thing, lifting off and exposing these little orange active sites. Myosin grabs a hold of the active sites and pulls. Okay? So repeat. Calcium binds to the blue thing, troponin. That lifts tropomyosin, the purple string thing, And then little orange sites here are active sites, which the myosin fingers, heads grab and pull. Okay, so that's what it looks like. Now, it's more of a process than that. Have you ever played tug of war? When you grab the rope and you pull, would you pull the other team in if you only pulled once? Or do you need to let go of that rope, grab further down, pull again, let go of that rope, grab further down, and pull again? It's a repeated action of grabbing, pulling, letting go, grabbing, pulling, letting go. That all takes energy. So when myosin grabs a hold of actin and pulls on it, it uses up the energy molecule called ADP. It's gone. So in order to let go, we'll watch the cycle again. ATP has to come in and break the cross bridge because there's no more energy for that to happen. ATP comes in, knocks myosin off, and in breaking that cross bridge, the ADP loses one phosphate and it becomes ADP. Okay? ATP breaks the cross bridge, becomes ADP, then it can grab and pull. ATP comes in, breaks the cross bridge. Myosin now is ADP, can grab and pull again. So it keeps going on, it keeps going on. So here's the YouTube video for that.
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head.
So ADP
cycle of movement
is needed to grab and pull on actin, but then ADP is gone.
Myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head.
And that's it.
This causes the actin myofilament to slide past the myosin myofilament.
ATP is going to swoop in,
is released from the myosin
break
head
the cross bridge.
The bond between actin and the myosin head
break.
Is broken when an ATP
it goes down one energy level to ADP. It goes in the cocked and ready position. It's ready to grab and pull again.
Stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
Over and over and over until the muscle's contracted. Okay. All right, got it? Good. Okay, so let's go back to, let's talk about rigor mortis, right? Now, at the time of death, when cells die, sometimes muscles at the point of death go through agonal movements, little contractions. And so that means there's going to be some cross bridges between myosin and actin at the moment of cell death. And at cell death, cells aren't producing any more ATP. So whatever cross bridges remain between actin and myosin, they can't be broken. So those muscle fibers remain in a contracted or tonic or muscle tetany state. So at the time of death, without ATP, the muscles lock up and the whole body goes into full muscle tetany about 12 to 18 hours after death. Now, 36 hours after death, the body relaxes again. It's not that ATP has been magically created. It's that actin and myosin filaments are starting to, well, decompose and the filaments are falling apart. And that's what's happening with that. All right. So without ATP, cross bridges can't be broken. Okay, let's continue on. So here are some factors that influence muscle contractile force. Because I've been talking about muscles contracting and shortening, but that's not the only types of muscle contractions there are. When a muscle contracts and it shortens, as this right-hand image shows, that's called an isotonic contraction. However, when you do isometric exercises where you're putting equal force from this hand against this hand, and really nothing is moving, okay? Your muscles are generating tension, but they're not shortening. So I've got this guy lifting a boulder. Well, he's not lifting that boulder anywhere. He can put pressure on that boulder and his muscles will have tone, but they don't contract because he's not able to push that. The mass is much too great for the force his muscles can generate. So that's called an isometric contraction. Iso means same and metric means measure. It hasn't changed. Okay, that is a fantastic place to stop for our first lecture, and our next lecture will be available on YouTube. Okay, so let's stop the share, and we are good.